So yes, today I'm going to talk a little bit about the work that we are doing in Copenhagen. And as Urs nicely mentioned, we are mainly working with the protein self-assembly. And in the title here, I want to highlight a little bit the broad interest that this kind of topic has been having in the last years, because there are several aspects that we should consider the moment we talk about protein self-assembly. So basically, if we would like to really synthesize where in which field protein self-assembly is specifically important, I would identify basically these three main fields I'm showing you in this slide. First of all, actually, protein self-assembly is it's a big problem. At the moment we go, we try to optimize the development of protein drug products. Indeed, this is an unwanted process in pharmaceutical production, because if you have a protein drug product, you want it to be stable, but this is not always the case. So there is a lot of interest in trying to understand what kind of physicochemical parameter, and also, for example, the material vials and things like this, how can affect the overall stability of the solution. So there is a lot of interest in trying to understand aggregation in this setting, and also try to characterize the particle formed within this formulation. But even worse is actually the connection that protein self-assembly has with neurodegenerative disease, as Alzheimer and Parkinson disease, since this kind of aggregated protein can be actually found in the brain tissue from Alzheimer's and Parkinson disease patient. So there is also a lot of research going on into this direction. But on the bright side, on the good side, in the last 10-15 years, there's been a growing interest in protein self-assembly, in controlled protein self-assembly for the realization of nanomaterials for different applications that go from drug delivery to also biosensing and tissue engineering. Actually, the team in Copenhagen, with my team in Copenhagen, we are trying to touch all these different aspects. I would like today to give a little bit an example of the approach we use to study protein self-assembly and the kind of techniques we use. And hopefully at the end of the talk, I will also give you a little bit of information about some recent results we got in the field of nanomaterials for application in drug delivery. So probably most of you are aware about this sort of general scheme about protein aggregation, but I think it's useful to point this out to in terms of in living system, what we have is the synthesis of a protein, and then this very well-known process that is bringing this linear chain to a three-dimensional structure that is called the folding process. But as you can see in this scheme here, along this pathway, you can also experience a series of other events that can generate different from amorphous, highly disordered species, as I show you up here, to very ordered structure like crystals, fibers, and oligomers. And then there is also a third class that is called this amyloid class that has some similarities with the highly ordered crystal, but still keep some of the properties of soft matter material. So actually, my team over the years has been working a lot in regard to this class of material, this amyloid materials, and the very well-known material, amyloid material, are these elongated fibrils. This kind of process happen, what are the driving force for this process, and if it's possible also to actually obtain different structure other than fibrils. So we came up with of course, all the work we do is actually initially in vitro. So it means that we start from a specific protein formulation, and we consider all the possible parameters that can affect this aggregation process. And that goes from any details about the handling of the solution to the kind of vial you use. But of course, the parameter that we usually change and we usually used to induce aggregation are actually stress factor as temperature and time, and of course to change the interaction among between protein in solution, we used to change a lot the solvent condition like salt, pH, and addition of several different co-solvents. So the idea is that what I'm going to show you from now on is actually results from in vitro aggregation that is obtained by changing mainly these two sets of parameters. So a few years back, we came up with this picture about protein aggregation, in which, of course, we start from a native sample, and then depending on the different interaction, we can actually end up not only with these amyloid fibrils I showed you before, but actually if we move the net charge on the protein and then we apply our stress condition, we can actually explore a quite broad number of different structures. And the two main ones that we've been focusing on in the last year are these two species. This species here, they're called particulates, that is in the range of microparticles. They are very dense. And these are the species that is a sort of core shell structure that are called spherulites. So we've been doing a lot of work trying to understand what are the driving force ruling this aggregation process. So what I'm going to do today is actually giving two examples regarding the study we generally do and the approach we use to understand the formation of these two structures, spherulites and particulates. And I will start actually with this one. And I think it is actually important to get a better idea, a little bit of a picture of what we are talking about. This one is, for example, an environmental scanning electron microscopy image of these spherulites. And actually, we, on purpose, we break the structure to reveal a little bit the internal part. As you can see here, you can actually identify a lot of directionality in the internal part. So this sort of radially elongated material in this shell. And this one is actually confirmed the moment you work with cross-polarized microscopy, in which you can actually identify specific part and specific directionality within the structure. And this method is quite useful if you want to identify right away the presence of this species in your sample. So we use quite a lot to actually be sure that the material we are talking about is actually related to this species. So this observation, together to a series of other experimental data, brought to the idea that this kind of structure are basically composed from a dense core, and then we have some protein material that is radially elongated from this core. Quite interesting, this is exactly the same species are found in brain tissue from patients affected by Alzheimer or Parkinson's disease. This also brought this diversity in morphology to some kind of attract some kind of big interest in the community, because it looks like that we do not have only one specific form of aggregates that can occur also in vivo, but we can actually have a variety. And this one is very likely depending on the boundary condition, the environmental condition in which the aggregation process takes place. And also, it looks to be quite general, because it's not only related to disease-related protein, but also to some globular protein, also some pharmaceuticals. So it looks like being a general state for protein, depending, of course, on the environmental condition. So now I will show you a specific example on how we tackle this question, how this aggregation or this aggregation pathway are activated. So here I'm going to show one example one example on how we studied this system. Actually, what we do here is actually using a very classical biophysical setup in which we select protein-protein interaction by adding specific co-solvent. So the setup we have here is basically using a biopharmaceutical like insulin, and we use the generic low pH condition and high temperature to induce the aggregation, but we do it in presence and absence of ethanol. The reason why we use ethanol is that ethanol is controlling the electrostatic interaction among the protein. Specifically, the moment we include ethanol in our solution, what we do, we increase the repulsion between the two proteins. At the same time, we also change the hydration shell of the protein. So this one for us was what we want to probe here is what is the role of the electrostatic interaction in the formation of this kind of species. So this is a little bit the setup. So we have a system that is in aggregation condition and another system in the same condition, but with the addition of ethanol. So we run the aggregation process and then we analyze the final structure we obtain. And to do this, we use a combination of confocal microscopy and also two photon microscopy with using specific dye. In this case, the red one is an hydrophilic dye, the cyan one is an hydrophobic dye, and the green one is indicating the amyloid nature of the system. So in the case of the system without ethanol, what we observe is actually that, the amyloid dye can actually stain the structure, but the hydrophilic dye cannot enter the structure, the spherulite structure. And here it's even much more visible if we divide the two contribution, in which we can notice that the red color is actually sitting on the surface of the structure. The hydrophobic dye is also coloring the entire spherulites, and actually you can appreciate this hedgehog fashion of this structure. The moment we move to the spherulite that are generated in a different electrostatic environment, what we notice actually is that in this case, the hydrophilic dye is actually able to get within the structure. So that means that we have already here a very big difference in the physicochemical property of these two materials. And it's even more pronounced the moment we look at the hydrophobic staining, in which we can see that the structure is much more compact now. It doesn't present the classical hedgehog arrangement. So that one was actually quite interesting for us, because by slightly changing the electrostatic, we actually generate a huge difference both in the physicochemical property of the material, but also in the morphology. Then the next question for us was, okay, is this somehow affecting also the mechanical property of the system? So to test this, we also use infrared spectroscopy, but we use in a setup of high pressure. That's called high pressure FTIR. So basically the idea is that we have our sample and we pressurize it, and then we look how the infrared spectrum is changing upon pressure. So basically what we want to do is looking at the spectra here that I show is just, this one is the native insulin spectra, and this one is actually the spectra out of aggregated insulin. And this peak here is the very well-known beta sheet peak that is indicating that we are forming amyloid structure. So generally, once we have aggregation, we have a transition from this spectrum to this spectrum. So the idea was to probe how this peak was changing during the pressurization cycle. So we did the experiment, of course, for the two kind of materials. And what we notice is that in the case of absence of ethanol, the moment we pressurize the system, we actually change slightly the infrared spectra. This one is the signal coming from the beta sheet. It's changing a little bit. At the moment we release the pressure, it's not able to recover the initial signal. Completely different story is in the case of 40% ethanol. So the system that was much more compact and able to incorporate hydrophilic dyes. In this case, the moment we pressurize, we go from this pink spectrum to this blue one. But what is interesting, the moment we release the pressure, actually the spectra is kind of coming back. So this one is again showing that in one case, we have a kind of hard material that is difficult to stress mechanically. And in the other case, we actually have a system that can be easily stressed, but at the same time, once the pressure is released, it's actually coming back. And again, this one is giving us a flavor on how we can control a kind of broad range of property of the material, going from hydrophilicity, hydrophobicity, but also to the mechanical property. Then, and this is some work we did recently, we're talking about quite big particle. As you probably saw from the scale bar in the previous images, we are talking about species that can reach several tens of micrometers. So one of our questions was, is this structure homogeneous, this secondary structure we observe homogeneous along the entire spherulites? So what we did actually is selecting a specific spherulite, and then we use a micro FTIR. So basically we do exactly the same experiment I showed you before with infrared spectroscopy, but in this case, we actually have a quite good spatial resolution. So we can actually focus the beam on a different point within the structure from the core to the external part. So we did this experiment, we detect different FTIR spectra, and we obtain what I show here. So the first observation we had is that overall the entire spectrum decrease when we move from the core actually to the external part. This one is actually not surprising, because if we consider the way we deposit our sample here, when we go from the core to the edge, actually what is the beam is experiencing a different amount of material. So it is expected that the moment we move from here to here, the total concentration of the material we probe is actually changing. So this one is not surprising. But what is also interesting is that the moment we normalize the spectra, we can actually see that this signal here, that is the beta sheet signal, is actually changing while we move from the middle of the structure to the edge of the structure. Actually, if you do a quantitative analysis of this, you will see that when we move from the core to the edge, we have an increase of the beta aggregate structure, while the alpha helical, the native structure, is decreasing. And again, this one is giving us an idea on how complex is the analysis of this structure and how difficult it would be to actually analyze the different contents of structure along the aggregates. So this one means that it's kind of impossible to evaluate the overall structure of this kind of species using bulk experiment, but we need to go to more detailed and high resolution experiment to reveal the real nature of these spherulites. So all this has been done all I show so far has been done a pH away from the isoelectric point of the protein, and where we observe the formation of this structure. So what we were focusing a little bit more, it was about the formation of a different kind of material the moment that we move a little bit closer to the isoelectric point of the protein. So the moment we move close to the isoelectric point, basically the charge on the protein, on the single, or the average charge on the single protein molecule is almost 0. So that's actually why we start also investigating this material here. And I will show you an example, and again, it's going to be on a model system that we chose to be a Hegon lysozyme, and we work at a pH that is 8.5. It's very close to the PI of the protein, and so we use again specific stress condition, and we use temperature to trigger the aggregation. So the first thing we notice is that the aggregation is happening in a time scale of minutes, that in the field of protein aggregation is a quite fast process, I would say. But what was interesting was the analysis and the comparison between the native state, so the protein structure at the very beginning of the process, and the aggregate structure at the end. As you remember before, I showed a huge change between the native and the aggregates. And actually, it's not the case for this kind of specific aggregate. Actually, what we can see, it's only a small change in the area of one, in this area here that is representing intermolecular beta sheet, and also a little bit of change in this area here. But the change is not massive as we observed before. And of course, if you do a morphological analysis, you will see these very compact microparticles, and the size, it's quite different compared to the one I showed you before. We are talking about here 1 to 2 microns microparticle. So one of the questions we had, okay, we have a very small change, we have this formation of particle, what is really happening during the kinetics? So to do this, we use a circular diagram that's is actually helping to probe the evolution of secondary structure during the kinetics. Actually, we did the experiment over time, and as you can see here, the spectra, this specific shape of the spectra is indicating mainly an alpha helical secondary structure. And this kind of spectra are actually kind of similar over time, but the only difference is arising from this signal here around 230 nanometers. If you look a little bit in the literature, you will find out that for the lysozyme, this signal is very much related to the environment of tryptophan 180 that is located here in red. So one of our hypotheses was, okay, something is going on around this region. So, and we also know that in lysozyme there is some sort of flexible domain here that is actually can be quite flexible and quite can be quite much affected by the environmental condition. So to check if this was the case, what we did is using the property of tryptophan to be fluorescent. So we can get fluorescence out of tryptophan, and what we did was this measurement of the fluorescence over time. As you can see here, we also have a quenching of the fluorescence. That means that we have a sort of opening up of the protein that is exposing the tryptophan to the bulk water, and this one is generating this quenching that we observe. So then the nice question was, okay, can we identify these intermediate states that is bringing to the formation of the final particle? And to answer this question, we use small angle X-ray scattering. And of course, I cannot give you a full overview on the technique, but I would like to give you at least the flavor of what we can do with this approach. So first thing, we work in solution. So we actually work in exactly the same condition in which we are carrying on the aggregation. And the nice things about this approach is that if you shoot, for example, new X-ray scattering here, you actually collect a specific on a 2D detector, a signal that can be converted in basically a 1D plot in which you have the scattering intensity as a function of the angles. So this kind of signal here contains a lot of information about the overall shape of your system. And it is quite suitable for systems that are in the range of 1 or 200 nanometers. And this exactly fits quite well with most of the protein in solution work that we do. So what we can extract here, it's after a quite advanced analysis from this 1D plot I showed you before, you can extract what is called the fair distribution function that is giving you an idea about the significant distances in your sample. And out of this, through some advanced analysis that nowadays can actually be done easily with some very well-tested software, you can actually obtain the envelope and the shape of the molecules you have in solution. So that's exactly what we did to identify this intermediate state. So that's a little bit the question mark we had, and then we managed to isolate this species here to run X-ray scattering experiment, and then to analyze the data and recover the envelope. And what we obtain is actually this green surface that you can see here. And on top of this, I actually overlap this native insulin lysozyme structure. As you can see, there is a difference between the native structure and this intermediate one. And specifically, the difference is in this tail here. And it's quite interesting because this one is actually perfectly matching the potential flip out of this flexible domain I mentioned to you before. So that's somehow confirmed this idea that this domain is just getting out of the main core of the protein. This one is also exposing these beta sheets that you see here. And we ended up also to hypothesize that the moment we have this beta sheet coming out of this structure, we can actually have interaction between beta sheet among different molecules. And from that point on, we can have the formation of the microparticle. So again, this is another example on how we deal with this problem of the understanding the kinetics, understanding what are the driving force bringing to the formation of these aggregates. I will just the very last part of the talk regarding a little bit some positive aspect of self-assembly, I would say, and it is some work that we start recently on controlled protein self-assembly. As I showed you before, electrostatic and it's actually ruling a lot of the reaction we look at in bulk, but actually we can also use it in a more controlled way, and we use a quite well-established approach to form protein self-assembled material that is called electrospinning. I think most of you are familiar with it, but the concept is quite simple. We have a protein or polymer solution in here, and then using a pump, you expose the solution within, in a area where we have a specific electric field, and then you have solvent evaporation. Then you can actually collect your solid material on this collector, and you obtain your fibrils. So we didn't invent anything regarding this, but our idea was to actually go for a material that was a protein material that was able to be formed without the addition of any inorganic component or toxic component. So our dream was actually to obtain a material that was almost completely formed by protein. And to do this, we actually decided to work with alpha-lactalbumin. It's coming from milk protein, but there's a lot of quite nice property that, and also some application in the field of drug delivery, but also of interest in nanomedicine and also for the targeting tumors. So the idea was to create this material out of this protein. Actually, the first achievement from this work that we started a few years ago was actually to be able to electrospin a protein solution. One of the things is that this solution, these fibrils are obtained from water solution. So we didn't add anything other than protein, the water, and a small amount of PO that is a carrier polymer. So the beauty of this system is that actually you can easily tune the size of the nanofibers, but by just changing the protein concentration. And we actually can explore a range that goes from 300 to 700 nanometer. And also, of course, these nanofibers, they keep one of the property of alpha-lactalbumin that is highly hydrophilic. So the moment they touch an hydrated environment, they dissolve immediately. But we also managed to have some post-treatment of these fibrils that, of course, the balance was to keep the biocompatibility of the system, at the same time improve the water stability. And we obtain it by using glutaraldehyde vapor. Actually, this is a little bit how it looks like. So here is the fibrils without any treatment. The moment they are exposed to GTA vapor, they actually show a little bit more entanglement. And the moment they are in water, they do not dissolve, and they actually keep more or less the same morphology of the network. What is actually interesting is also that together with tuning both the sides and also the water stability, we can actually have a system that they have a completely different mechanical property. Here is the Young modulus for each of the system. As you can see, by increasing the concentration of alpha-lactalbumin, we actually decrease the Young modulus, and the moment we cross-link the system, we actually increase again. So as you can see, again, there is quite a large room for tunability for this system. Of course, another test was what is the effect on different cell lines, and the morphology of the cell upon exposure to this system is actually not changing that much, and also the cell viability is actually telling us that the system is actually good, is not creating any issue from a biological point of view. And then in this first study, what we also had was a kind of proof of concept about the possibility to incorporate drug into it. And actually we had this model antibiotics that it was ampicillin that was successfully embedded in this protein nanofibers network. And actually we tried to look how the release of ampicillin was in looking at the inhibition of bacteria growth. And also the system was working quite good. Here is some results about the inhibition area in different phases of the bacteria growth. But what was interesting is that at the end of the process, we actually managed to kill the bacteria since we didn't observe any bacteria grow. So it means that actually the system, or at least this ampicillin-based ampicillin incorporated in this protein nanofibers, were actually able to avoid the presence of persister at the end of the treatment. So we were quite excited about this material, and we wanted to use this property I mentioned you at the beginning, that these nanofibers that actually dissolve in water quite easily. So, and we thought about that this can be a good property if we one wants to develop a system to for the release of nicotine. I mean, there are a lot of systems for this, what is called the nicotine replacement therapies, that generally should help people to stop smoking. But one of the problems is that most of the systems, they are not able to release nicotine in a very fast way, in a very localized way. So our idea was, okay, can we incorporate nicotine within our system and try to see, to compare the performance of the system to some of the market products. So that's actually what we did. And the first achievement was also to be able to incorporate nicotine, a quite decent amount of nicotine into the fibers. And that was achieved. And as you can see here, the nanofibers are not that affected by the presence of the nicotine. And then the first thing we did was actually to check the release in the possibility to release it in vitro, and we compare with two market products that you can see here. As you can see, our system is actually able to release nicotine quite fast and in a quite considerable amount. So we were quite happy about this, and then we, of course, move into the standard check on cell viability. That was also quite satisfying for us. And also we moved to, we also look at the permeation and transport towards epithelial cell. And also in this case, we could see a good transport from our nanofiber system. So what was actually quite surprising is actually the ex vivo study we did. So we took our system with our nicotine, and then we exposed to buccal mucosa, and then we look if there was any delivery of any permeation of nicotine. So we followed this by MALDI spectroscopy, and here is the two systems we investigated. So this one is actually how the tissue looks like, and then it's here we have the application of nanofibers. In red, you can see actually the nicotine. As you can see here, the moment you apply the nanofibers, we have some permeation and some nicotine is going through. If you have just the solution on top of your tissue, we actually do not observe any transport, any permeation of nicotine. And this one is also nicely represented in this scheme here, in which we have the cumulative nicotine permeation for both the condition. So we were quite happy. And then one of the things we think one of the mechanisms that we think is at the basis of this quite large permeation is that we have a system that is actually able to concentrate quite a lot the nicotine, but at the same time, the moment it's in contact with the hydrated surface, you can actually have an immediate release that is highly localized. That is exactly the problem that some of the therapies are encountering nowadays. So I think I'm now approaching to the end of the talk, and then I would like just to summarize a little bit what I showed you before. So I hope I could show how important is the formulation in creating a different variety of aggregates, and how it's possible to study this different structure. I show you two examples that are a kind of the two extreme the moment you talk about protein aggregation. One is this spherulite, this conversion from native to beta-rich structure, and also this peculiarity about this spatial heterogeneous structure that they have. And on the other side, we have these microparticles that they have minor change. They show minor change compared to the native structure. It's quite peculiar. And of course, this idea of creating this sustainable and green protein material for drug delivery, that is something we are working on. Of course, we are nowadays working and keep on working on this knowledge that we have been building in the last years. And specifically, we work still on the occurrence identification of particles, and there are several good reasons to study this, because the different characteristics that we can analyze with our approaches are quite important to identify which features are actually related to potential immunogenic potency of this kind of particle. And of course, we always look with curiosity about the implication of the different morphology in human disease. But something that is also come up in our work nowadays is also the possibility to use this system as a potential carrier and a potential delivery system. And of course, there is also some ongoing work on engineering more advanced protein nanofibers, and hopefully we will have more results in the coming years. I think I'm done with this part. I just need to, the most important slide probably is the acknowledgement, and then of course it's quite important the support we get from the drug delivery biopharmaceutical group we are in. And of course, a special thank you to the team working on protein self-assembly, and specifically today I presented work from the scientists that are highlighted here. But of course, this work cannot be possible without good collaborators, and we have a long-standing collaboration with the University of Palermo regarding the fundamental understanding of protein self-assembly. And while for the more applied part I presented here, it's a joint collaboration between the University of Copenhagen, Exeter, and initio. And of course, I need to thank all the sponsors, all the foundations that are actually allowing us to perform our research. And thank you very much for your attention.
Thank you very much, Vito. Very interesting talk. You actually might refresh your video because you've just turn it on and off again, because we can't see you. The first question will be by Marcel Bali, because he wanted to ask a bit more about the immunogenicity of these materials that you work with.
Yeah, I should say that I have Zoom frozen, so I hope it's fine not looking at my face, and I can listen properly.
Yeah,
yes, okay.
Okay, hi Vito. I think I'll keep it short because I think you touched on it at the end of your presentation. But I was just commenting to Urs about the fact that these guys are probably going to be highly immunogenic, and I think you've mentioned that, but you also implied that there's a way of controlling immunogenicity, and I was wondering if you could speak to that.
Yeah, I cannot tell that much because it's an ongoing project with a company. So that's, but what I can tell you is that I think this is quite, it's quite a debated point, because I mean, we don't know if they're immunogenic, but of course, what we can do is characterize this particle properly in a way that we can identify the different features that characterize this particle. Then it comes the experiment regarding immunogenicity. Then the big thing is to relate the level of immunogenicity that we register with the specific feature of the particle. I think that's the key point that we would like to address. We do not have results yet, but and also it's difficult for me to also predict if any of these particles can be more immunogenic than another. But I think before going and understanding immunogenicity of this particle, we need to go towards a more careful and high resolution characterization of them.
I'd agree, thanks.
Okay. And one other question that several people had is about, you know, what are really the biggest benefits of using this protein self-assembly? And they were specifically mentioning blood factors, you know, coagulation factors or insulin. What do you see there, the biggest benefits?
Yeah. Yeah, it is a good question, and it's a good question for myself because as you could probably see from the presentation, we are entering a little bit this field of material from protein self-assembly quite recently. But one of the things that I've been seeing also, it's a clear pattern in all the projects we are having, is also not only the possibility to use the protein self-assemblies as material, as a way to produce the material to carry something, but actually we can use protein self-assembly to create a sort of new formulation for a protein drug. I think it is quite relevant because in some cases we see some benefit of having a protein not in, let's say, monomer native state, but rather in a more, let's say, oligometallic state, but still keeping some of the property of the native state. So in that sense, it's this idea of using the material as a depot of actually the active biopharmaceutical. So that one is, I think it's a big advantage that I see in using this technology. But then, of course, I think there is a huge literature regarding also the use of controlled protein self-assembly. Is a little bit more protein engineered there, in which they build the material with the specific structure they want, and because there is a more specificity towards binding with some of the other active ingredients that they want to deliver. So that's a little bit the way I see it in terms of advantages.
Okay, thank you very much. I think we have to stop there, unfortunately.